In this video, we're going to continue our description of action potential by talking about propagation of the action potential down the axon. So in the previous video, we described the generation of the action potential up through the events that occur immediately following depolarization at the axon hillock. And so what we're going to do now is just we're going to redraw the axon, and we're going to just sort of highlight the sodium channels. So instead of drawing all of the channels that would be present, we're just going to draw the sodium, but we know that the other ones are still there. And I want to talk a little bit about how the action potential actually moves down the axon and why it moves only in the forward direction. So we know that there's an abundance of sodium outside the cell, and then there's a small amount of sodium that's accumulated inside the cell. So we'll just kind of revisit where we left off last time. We have the sodium that flows in through the voltage-gated sodium channels, and it accumulates right sort of in that vicinity. And we also know that there was some sodium that was concentrated at the axon hillock that caused that depolarization to occur. So as that sodium moves away from the other sodium ions, it moves in the forward direction down the axon, it will depolarize the next sodium channel. So that sodium channel will allow influx of sodium ions, and as those sodium ions come in, they'll move down the axon and depolarize the next sodium channel, and that sort of process continues all the way until we get to the end of the axon where we see those calcium channels. So I just want to highlight a couple of important components of this process. One is we know the sodium wants to propagate, move outward in all directions. So if we think of the sodium moving backwards, we know that it really isn't going very far because there's an abundance of positive charges behind it. But more importantly, the voltage-gated sodium channels that were open when that sodium came in, those are now out of commission. They're in their absolute refractory period. So the sodium has no effect moving back towards those sodium channels. So the sodium is going to move in the forward direction, and it will depolarize those sodium channels that are forward. And also, while the first set of sodium channels are in their absolute refractory period, it's also important to know that during this time, the potassium channels are open, sort of adjacent to those sodium channels, and they are allowing the efflux of potassium. So as the potassium is moving out in association with that first segment of the axon, the second set of sodium channels are open and the sodium is rushing in. That sodium will create a local current to depolarize the next set of sodium channels, and then as the sodium rushes in that next set, the second segment of sodium channels are in their absolute refractory period, and the potassium is also leaving the cell. So it's very important to think of these events as simultaneous, because that the propagation down the axon is really all about local current, and the fact that the action potential can't travel backwards because the previous segment is in the absolute refractory period. But that doesn't mean that we can forget what is happening with the potassium channels and then also the sodium-potassium pump. Those things are happening in a sequence: sodium, then potassium, then sodium-potassium pump for each one of the sodium channels that we have represented in this particular portion of the axon. So this method of propagation is referred to as continuous propagation, and it's what happens when an action potential travels down an unmyelinated axon. So we mentioned myelination in a previous video, and now we know that we didn't draw the myelin in there, so this is going to be continuous. It's important to understand that this process is a positive feedback process. As sodium enters in the voltage-gated channel, it moves in the forward direction, causing more sodium channels to open, more sodium to come in, further progression of the action potential forward, more sodium channels open, more sodium comes in, and so on. So it's our first sort of real example of positive feedback in this course, but it's important to keep in mind, at least to understand that positive feedback is a normal process in a lot of ways in the human body. All right, so now let's take a look at what happens when we have a myelinated axon. So we highlighted the fact that there's a difference in the behavior of the action potential. So here we're showing our myelinated axon. We have several segments of the axon are myelinated, and then we only have sodium channels in the gaps in between those myelin sheaths. So of course we'll have the sodium, of course we'll have the potassium and the channels and the sodium potassium pump, but we're just showing the sodium in this example. So the sodium is going to be present at the beginning of the axon. It travels down to that first set of sodium channels, causes the sodium channels to open, and then there's a long space before there's another sodium channel. They're not as closely packed together. We don't see more sodium channels until we get to the next gap in between the myelin sheaths. And so what happens is the sodium actually diffuses along the inside of the axon, like along the membrane, until it gets to that next set of sodium channels. So this process is referred to as saltatory propagation as opposed to continuous propagation. And there's a couple of important points to know about saltatory propagation. First, it only occurs in myelinated axons, but those myelinated axons include the axons of the CNS and the PNS. The myelin functions as insulation, so there's no sodium channels where the myelin is. And as a result, the channels are localized to the points in between the myelin sheaths. So I just want to mention that where the myelin is, we call that an internode, and when there's no myelin, we call it the node of Ranvier. So we're showing here 2 nodes of Ranvier and then 3 internodes. And of course, there would be as many as there are sort of myelin sheaths along the axon. The next important question is, why do we bother with myelination? And the reason is because there's an increased rate of propagation in myelinated axons. It's a dramatic difference. So basically, the rate of diffusion, as we mentioned, from node to node, so from set of sodium channels to the next set of sodium channels, is significantly faster than trying to open every single sodium channel along the axon. So if we reduce the number of channels, insulate the axon, and then have the current diffuse along the membrane a longer distance, we actually can accomplish delivery of action potential to the synapse significantly faster. So that concludes our explanation of action potential propagation, and in the next video we'll talk about the events that occur at a chemical synapse. So that's all for now. Thanks for watching.